Hello everyone, my name is Binay. Welcome to my channel Lifecycle. Today we are going to learn about erythropoiesis. Erythropoiesis happens in bone marrow in adult, but in embryonic stage it happens in yolk sac. Suppose this is in embryonic stage, it happens in yolk sac. So when it becomes fetus, suppose this is the placenta, this is the fetus, it happens in liver. So erythropoiesis happens in fetus in liver, and then when the fetus develops and before the birth, these hematopoietic stem cells, as in my last video I have described about the hematopoiesis. So hematopoietic stem cells move to the bone marrow, and before birth most of the hematopoietic stem cells move to the bone marrow, and after birth these hematopoietic stem cells present only, hematopoiesis happens only in bone marrow. We'll discuss about adult erythropoiesis. The first step in erythropoiesis starts with erythropoietin. Erythropoietin is an important hormone which is secreted from kidney. In kidney, lack of oxygen is sensed by some specialized cells and mainly by transcription factor HIF. This is hypoxia inducible factor. Hypoxia inducible factor. So this transcription factor produces EPO, erythropoietin, and this erythropoietin moves to the bone marrow. And as I said in my last video, from hematopoietic stem cells, hematopoietic progenitor cells produced, and from hematopoietic progenitor cells there is another kind of progenitor cell that is called common myeloid progenitor cells. From common myeloid progenitor cells, myeloid erythroid progenitor cells are produced, and from myeloid erythroid progenitor cells, erythroblasts are produced. And in this stage, this erythropoietin works on these cells to differentiate into erythroblasts. And erythroblasts are differentiated into early erythroblasts and late erythroblasts, depending on their function. Late erythroblasts. In early erythroblasts, nucleus is there, and in this the ribosome synthesis happens. There will be so many ribosomes in early erythroblasts, and when it becomes late erythroblasts, the hemoglobin synthesis happens. There will be so many hemoglobins, and after that they differentiate into reticulocytes. In these reticulocytes, these reticulocytes are very much similar to the fully formed or differentiated mature erythrocytes, but these are the reticulocytes, and these reticulocytes contain so many ribosomes, and after forming the reticulocyte, they usually most of the reticulocytes stay in the bone marrow, and some of them move to the blood. This is the reticulocyte, and reticulocyte usually stay in the blood stream for around 24 hours, and after that they differentiate into the. Before forming reticulocyte, this nucleus is lost here, and from late erythroblast, nucleus lost, and then it formed to reticulocyte, and from the reticulocyte, and this nucleus is destroyed, and this reticulocyte comes to a bloodstream. From reticulocyte, erythrocytes are developed, and these erythrocytes or RBC, they look like biconcave. So, they look like this. From side view, they look biconcave, and they have huge surface area. So there is a reason why they lose the nucleus. This is the only cell in our body which does not have nucleus. So why they lose their nucleus? Because after losing their nucleus, they have huge area in the cytoplasm which can contain maximum number of hemoglobin, and each hemoglobin can bind to 4 molecules of oxygen. So that is why the more the hemoglobin, the more oxygen they can contain. So that is why they lose the nucleus, and they have membrane which can help in oxygen exchange with the cell or tissue. So after becoming RBC, they move in the bloodstream, and also they go to the lungs to exchange with oxygen. They get oxygen from the lungs, and then they move to the bloodstream. They move in the bloodstream and move to the tissue area, and in the tissue or from the cell they exchange CO2 with oxygen, and then they carry oxygen to lungs, and this process continues. And now coming to the destruction process. So usually the life cycle of one RBC is around 120 days. So during this process, they sometimes they rupture their cell wall or membrane, plasma membrane. So after the becoming old or after the destruction, usually the macrophage in the spleen because macrophage has phagocytosis capability. So macrophage in the spleen or in the bone marrow, they can sense the old RBC or destroyed RBC. They phagocytose these destroyed RBC or old RBC, and they make, this is the spleen, and they make Fe, the iron, and biliverdin. This is from heme, and the globin part is converted to amino acids. And these amino acids again go back to the bloodstream and they make again some proteins. So this biliverdin is converted to bilirubin and is transported to liver. And from the liver, these bilirubin are transported or secreted out to feces or in the urine. Bilirubin is secreted out through liver, and what happens to the iron? Iron is transported back in the blood through transferrin. This is a transporter protein. This transfers the iron in the blood because iron is very reactive, so it needs a transporter protein to be transported. So this transferrin transfers iron to the bone marrow. Again, it produces hemoglobin and it helps in hemoglobin production. And this process of destruction of RBC is called Eryptosis. The production process, the differentiation process is called erythropoiesis, and the destruction process, the death process, is called eryptosis. This is all about erythropoiesis. Thank you for watching my video. Bye bye.